Hi friends, in the protein chemistry we have primary structure, secondary structure and tertiary structure. So in this video let us try to understand the importance of primary structure in maintaining the protein conformation as well as the function. So conformation means the structure, okay. So, the importance of primary, right. See, basically we all know that the proteins are made up of some basic units which are called as amino acids. Okay, amino acids. So, we have amino acids. Commonly available amino acids are basically 20 amino acids. So, your amino acid concept that particular subsection will give you the list of amino acids like arginine, methionine, lysine, leucine, isoleucine, etc., and you have totally 20 such amino acids, commonly available amino acids, right? So this 20 amino acids, they are generally arranged in a particular sequence, like say for example A1 to AN, and this N will vary from protein to protein, okay. So what is this actually? This is the amino acids which are linked or which are connected by a bond which is called as a peptide bond, okay. So friends, amino acids will combine to form protein. So we know this, or we can say proteins are made up of amino acids. So in this process, the amino acids will all get lined up, say A1, A2, A3, it goes till AN, okay, and the bond between two subsequent amino acids will be the peptide bond, okay. Now this length, okay, the length, so this is what is called as a peptide chain, okay, this is what is called as a peptide chain. So it is easy to remember why it is called as a peptide chain, because it is a chain, okay, that are linked by peptide bond, hence it is called as a peptide chain, okay. So here this length of the peptide chain, say this value of this N will vary from protein to protein. Say if you say insulin as an example, you can expect this N value to be around 51 or so. So this varies from protein to protein, okay. So one is nothing but the length of the peptide chain, okay. Length of the peptide chain is one parameter, okay, that play an important role, okay, right. Say for example you have protein P1, protein P2, so A1 to AN, here also A1 to AN, the N value here, let's say it is going to be 55 and here it is going to be say 60. So as I said, the length, okay, is going to define what P1 and what P2 is, because P1 is also a protein, P2 is also a protein, both are made up of amino acids. So here what factor that determines this? Here this is made up of 55, this is made up of 60. So 55 is unique for P1, 60 is going to be unique for P2. Right, hope this concept is clear. So length of the peptide chain varies from protein to protein. Say let's take you have another protein P3 that might have some 200 amino acid, P4 that might have some 500 amino acid. So one parameter as I have mentioned here is the length of peptide chain, okay, is one thing important. Clear? Say even though the length is going to vary from P1 to P2, the other aspect is the sequence. The other aspect is going to be the sequence of amino acid, like in what sequence the amino acid is getting aligned or arranged, right? Say for example, let's assume that P1 is also going to be 55 amino acid, P2 is also going to be 55 amino acid, then this protein P1 and protein P2 are going to be same? The answer is no. Because though the length is same, okay, as considered here, but the sequence of amino acids will be completely different, right? So two things that play important role in determining the protein uniqueness is going to be the length of the peptide chain as well as the sequence of the amino acids in the peptide chain. So these two factors actually determine the identity of a particular protein. Say if you are going to manipulate either of this, either the length or the sequence, then the protein identity itself will be changed, right? So you always say you have digits 0 to 9, okay, and we pick up this digits, say 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have totally 10 digits, and we try to put this in a particular fashion, okay, and we get a mobile number, right? So you have a length and you have a sequence. And if you are going to change, say you have a person X, okay, whom you are going to use this, dial this number and talk to him. And if you are going to change this either a length, say you add extra number, or you are going to delete some number, or if you are going to change the sequence, then what happens is you will not be able to talk to this person X. Either it is going to be an invalid number, or it is going to a person A, B, C is going to attend your call. But your purpose of call is to talk to person X will not be fulfilled here. Likewise, if you have 20 amino acids which are getting aligned in a particular length in a particular sequence, and if you are going to manipulate this, right, like you change here, instead of 93678, you are going to dial 93778. Okay, your purpose is not fulfilled here. Likewise, you have an amino acid sequence, say you have methionine, alanine, tyrosine, threonine, again methionine, and you have this sequence at this length, okay, and that makes your protein P1. Right now, if you are going to change any of this, say curtail the length or add the extra amino acids or change this sequence, say you're gonna instead of alanine, you're going to replace this with methionine. Okay, replace this with methionine, then you might end up with an useless protein or some other protein, but not your protein of interest here, P1. The same case with P2. Okay, so in fact, two things that determines an identity of a protein is the length of the peptide chain as well as the sequence of the peptide chain. Okay, hope you are getting my point. And again, if you are going to manipulate this, both the parameter, we end up with a other protein, sometime a totally a different protein, but not the protein of our interest, that is the P1 and P2 here. Now, you can understand from this information that just from 20 amino acids with these two parameter of different length and different sequence, you might end up with millions and millions of protein. As just from 10 digits, you have n number of mobile number getting generated, but 20 amino acids you can have with a different sequence and different length, you can have a variety of protein in this living world, right? Friends, this thing, what is that? This length and sequence with which the peptide chain is formed, is what is referred as the primary structure, and this in turn is going to evolve into a secondary tertiary structure which finally gives the functional protein, right? So now you get to know what a primary structure is, and manipulating the primary structure by altering these things will totally changes the protein to a different thing. Okay, so hope now you have understood the importance of primary structure. Okay, in another video, let's meet with another interesting aspect. Thanks for watching.